Hello. Um, this is one heck of a chapter. In many fundamentals textbooks, these are two chapters. And I'm also cursed with a lot of information about this topic. I, I taught the course in psychology, sensation, and perception for quite a while, and I did enjoy it. And the opening vignette is relevant to me because historically I was a pain researcher and very interesting uh, condition that Gail has. Um, and sometimes, pain is there to help you. It's there to tell you when you're doing things that are damaging to the body or your body's being damaged. And people who don't feel pain can get themselves in a lot of trouble. So the book opens up with an overview of what is sensation, what is perception, what about the receptor cells, how many kinds of sensation are there, and more than we used to say in our oversimplified version of sensation. This table is good. The five most common: vision, touch, audition, olfaction, gustation. But if your vestibular sense ever goes bad on you, boy, you'll notice that. Some people get very dizzy because, well, there's different reasons for it to happen. But I've had a few bouts of intense vestibular problems, and boy, I don't enjoy them. Body sensation, proprioception, things we probably can't do, electrical and magnetic sensations. Whoop, somebody's yawning. And adequate stimulus, the type of stimulus for which a sensory receptor organ is particularly adapted. So the adequate stimuli are there in the third column of table 6.1. And this figure is also in my behavioral neuroscience textbook. And there's a cross section of your skin, and it's complicated in there. It can be quite complicated, and there's an emphasis on pain here, and not surprisingly, those are free nerve endings, and there are some other specialized receptors for vibration and touch and stretch, and they'll be dealt with a bit later. Labeled lines is an important concept. Which neurons are active. There's also sensory coding, and they can really do this. So you have a stimulus and you can record from peripheral nerves in real humans. And in A, you see that black line with spiky things. Those spiky things are action potentials. So there's a light touch where you get a burst of activity, and then the neuron slows down, and then you have a stronger touch where you have a longer burst of activity, and it still slows down, but it's higher than it was in A. So neurophysiology is important here. Action potentials are important here. Concepts of thresholds are important here. So there's a stimulus that's below threshold, you don't get an action potential. And then you get a stimulus that's stronger, it gets you to threshold, and you do get an action potential, probably more than one. So that's all good, reviewing some things. And this is an important concept that, and the skin is still good here. As an example, you have some neurons that have low thresholds and they have their own dynamic range, and then they kind of pass it off to medium threshold receptors and then high threshold receptors. So, to feel the full spectrum of that sensory system, you need to get information from all of those. In terms of the history of psychology, psychophysics was very important. And, it's the interface between physics, and of course, you can quantify different kinds of stimuli physically, and sensation and perception. And the absolute threshold is the threshold for detection, which sounds absolute, like yes or no, but don't forget these shapes up here. They're sort of S-shaped, and absolute thresholds aren't absolute. And there we go. So the neuron may respond a bit below what we call the absolute threshold, which we decided is the point at which there's a 50% chance of response or detecting the stimulus. And so absolute is not absolute. There's a range. And then we deal with just noticeable differences, JNDs. How different do two things have to be before you notice that they're different? And there's Weber's fraction. And that's not true for all ranges of stimuli, but it is it's a pretty good overview of what is typically true. Our absolute thresholds are pretty amazing. Vision, a candle 30 miles away, touch, the wing of a fly landing on your cheek, ticking watch 20 feet away, a drop of perfume, 3 rooms, and sugar, a teaspoon and 2 gallons. There are other animals that are better than us in many things, but our sensory systems are pretty impressive. And now you're getting into signal detection and signal detection theory, which, by the way, was like the biggest advancement in psychophysics in the last more than 50 years. And I want you to understand this to the extent you can. It's a bit confusing. It's also a bit statistical in terms of normal distributions and area under the curve. That table, you should know it. Outcome through detecting signals and noise. So the event, there's either a signal or there's not. It's S plus or S minus. And then you don't get to say you don't know. You have to say yes or no. And if you say yes to a signal, that's a hit, and that makes sense, like baseball. If you say yes to no signal, that's a false alarm. And that's not too confusing either. You turn in a false alarm, you think there's a fire when there isn't. You say no, there's no signal, but there was, that's a miss. Swing and a miss. And you say no, and it was no, that's a correct rejection. So I think you can see why that's important to sensation and perception. But this is also important to decision theory, which is important to many aspects of life and medicine and even the legal system, potentially. Although the legal system is not a big fan of using cutting edge science to make decisions. I'll show you some stuff on that in a bit. So there's noise, and then we add a signal, which pushes that perception over. So if the signal's not there, you have the red line. If the signal is there, the signal adds a constant to every part of the no signal distribution and shifts it. And then you get involved and you get to make a decision line, a criterion, where your perception of the world, if it's to the right of that dashed line, you say, yep, there's a signal. And if it's to the left, you say, nope, there's no signal. Of course, the distributions overlap, so you're going to make mistakes. So there's examples of the 2x2 table. You say no to no signal. That's good. You say yes to a signal. That's good. That's a hit. You say yes to no signal. That's a false alarm. You say no to a signal. That's a miss. So, look at this figure on this page and go back to that 2x2 table, and spend some time on that. It will help you understand. And this is real life. This is dating. This is whether you get a job, whether they think you have cancer. Mistakes will be made, but we do the best we can. And we have to look at the consequences of making a mistake. I tell you, you don't have cancer and you've got cancer, that's bad. And we don't want to do that. So we might move that decision line to minimize the likelihood of missing cancer. Of course, if the signal and no signal distributions are very far apart, you don't make many mistakes. But unfortunately, in real life, they overlap. They can overlap quite a bit. And then your decision line can make a big difference. If you were in admissions at Harvard or Yale, you would have your decision line way over on the right, because you'd have a lot of candidates, and you could move your decision line so the people you admit are all really good, and there's virtually no losers in your class. But if you're dealing with a school that's having trouble filling its seats, with the same data, they're going to move their decision line way to the left, and they're going to start taking in a lot of students who just can't handle it. And they'll make a lot of false alarms. Think about that. And okay, so I gave you a figure. I'm not assigning this, but I wish you would think about it a bit. So there's an organization I was a member of. They have a journal, the Psychological Science in the Public Interest. These are available free. And if you look in your textbook at who's involved in inventing signal detection, you'll see Sweats was there. Robin Dawes is dear to my heart. If he's still alive. He was a keynote speaker at one of our psychology conferences, and I don't know John Monahan, but psychological science can improve diagnostics. So this is not about psychophysics, but it's about decision making, and science can help with that. It can help with medicine. Physicians sometimes don't like it. Artificial intelligence and machine learning is important now. That's related to this too. So we want to enhance the accuracy of our decisions. And oh, look, I've seen that figure. So there's your, here you're dealing with normal and diseased, and you have a decision line, and this decision line is right at the mean, median, and mode of the diseased people. So you're getting, you say yes to half the diseases, but you say no to half the diseases. And then the non-diseased people, you still say yes to a percentage of them. And that's important to real life. Every time someone checks your x-rays out to see if you have a tumor, or you get blood work to see if you're in the normal range, these kinds of decisions are made. And there's a discussion that largely overlaps with what's in the textbook. And 4 illustrative diagnostic tasks, and whether a patient will engage in violent behavior, whether a radiologist has a woman that has breast cancer, or relevant to that, a male has prostate cancer, and how does signal detection play into that? And the bottom line is, it's good. I used to get my X-rays read up at the place that's near what used to be Toys R Us, which is gone, on the Dixon City Highway. And how important is the physician for reading the X-rays? Well, if you had your X-rays read at Penn, those physicians would do a slightly better job than the people here on average. But if you didn't let the physician make the call and fed it into a decision algorithm, like we've been talking about with signal detection, the X-rays taken here would be read as accurately as the ones at Penn. But if you had the Penn doctors feed their data into a decision algorithm, they would get even better. And we didn't cover this, and we won't cover this. It's important. It's important, but we won't deal with it. And then we get into the how we deal with decision-making theory and different kinds of goals and probabilities. This is too much. Don't spend any serious amount of time looking at this. But whether people are let out of jail on parole, predicting violence, if you do decision theory instead of experts, you get better outcomes. And experts like to be experts. They don't like, computers to tell them what to do. And here's the cancer, and this is relevant to the X-rays analyzed here or in Philadelphia. There's breast cancer data, and there are prostate cancer data. So, sensation of perception is a very important part of psychology. And signal detection was revolutionary when it first came into existence. But boy, it's relevant to more than just psychophysics. And then we have adaptation, largely the changes in the responsivity of the receptors, and eye saccades are there. Schizophrenics have unusual eye saccades. So there you go. sensation perception, labeled lines, absolute threshold, which, is a statistical concept, 2 by 2 table, Weber's fraction, J and D's, good stuff. Touch and pain, used to be my field. So there we go, looking inside the skin again, and now we're naming some of these things. Free nerve endings are the ones that are most important for pain and temperature. I speak for all the phonemes in temperature. It's not temperature, it's not temperature, it's temperature. When I was you, we used to mandatorily take a lab in sensation or perception, and we had a device that did this one. It really is pretty amazing. You just do the warm, oh, it's warm. You do the cold, it's cold. But you mix them together and yow, that's hot. No, it's not. It's an illusion. And then, different kinds of receptors have different receptor fields and different functions. And again, we can see these. We can take little biopsies and see these guys. And there's our old friend Aristotle. Aristotle's touch illusion. I've never done that one. I teach about it, but I've never done it. Kind of weirds me out. I don't know why. Not being able to feel pain is bad. I think earlier in the book, I complained that this little cell body wasn't there, but there it is. So there's injury and complex things happen in the skin. And by that, there's a cascade of events. It's complicated. And typically action potentials go in, they synapse on a secondary neuron in the spinal cord, and that neuron decussates or crosses and goes to the brain. And remember that sensation and motor systems are generally crossed. So there's a nice path for pain. Of course, that cell at the here, that would be multipolar. This is actually what that neuron looks like. It looks like a lollipop. This one is stylized. That's the multipolar. And congenital insensitivity to pain, generally not good for you. Some people seem to survive it pretty well, but others don't. And then there's this family who have individuals in it that are insensitive to pain. And now that we have DNA screening techniques, we have a very good idea of what's going on in these families. And the concept of alleles are important here. And the people who don't feel pain have KIP. And so that particular sodium channel, and that should remind you of the action potential, is required for nociceptors to produce action potentials to inform the brain about pain. When the channel doesn't work, no nociceptor signals reach the brain and there's no pain. Very interesting story, and it helps you review your action potentials. And don't forget the final, it's comprehensive. Pain illustrates there's a huge difference between sensation and perception. All right. We'll keep talking about pain. Let me add something for you. The puzzle of pain. This was important to me back in the day by Ronald Melzack. Unfortunately, now dead. But this was written for the general public, and it's old. And there's a newer book, The Challenge of Pain. You're going to experience pain directly or in your relatives during your lifetime, and you probably already have in terms of chronic pain. This is a very easily read and important book. Out of signing it, I think it's an important book. I think I have 2 copies of it somewhere. Last time I saw Melzack, he was walking alone down a median at neuroscience meeting oh no, that was Pat Wall. It was not Melzack at a neuroscience meeting. Melzack and Wall collaborated on a lot of research. And phantom limb pain is a weird one. And Ramachandran on the next page, I hope. Yes. So there's Rama's book. And I don't, clenching your fist and arcing it behind your back, I don't recommend it. It gives me an aching, creepy pain that sometimes takes a while to go away. I wouldn't try it. But there's the box that Rama, as he likes to be called. Oh, I shook his hand at Neuroscience one year. And boy, does he give good TED Talks. Really good. Ramachandran TED Talks. So here you have a mirror where the missing limb is shown in the mirror. And when this hand moves, that hand moves. And for some people, that can have a dramatic impact on the perception of phantom pain. And it's cheap. And nice discussion there. I recommend that you take a little time and watch a Ramachandran book on neuroscience. So in my book, this next figure is missing the parts of the eye. Now your book probably has it, but mine is missing it. So I've given you and me a picture that I can look at. So there's your cornea and your iris and your pupil and your lens. That's a good figure. And remember that the what's focused back here on the retina, oh, you can even see the fovea, that little dimple. What's focused on the retina is upside down and left to right. This used to freak out medieval researchers as they were taking breaks from arguing about how many angels could dance on the head of a pin. They were troubled that the soul, the mind, had to deal with a visual stimulus that was upside down and left to right. It troubled them. I am not troubled. By the way, most of the light bending that the eye does is with the cornea, not with the lens. The lens is for fine touch-ups. And we have photoreceptors, we have rods and cones, and there they are. And we have, there's another missing segment here, but I think that other figure will help with that missing information too. Now, what's weird about our visual system is it's not intelligently designed. Octopuses have intelligently designed eyes. Their photoreceptors are aimed at the incoming light. Our eye retina is in backwards. For the visual stimulus to get to our receptors, it has to go through all these cell layers. And that can fuzz things up. But of course, we can evolve mechanisms to sharpen it back up. So don't forget there are rods and cones. And there's a nice little table. 4 million cones, 100 million rods, lots of rods. Sensitivity, location in the retina, and cones are mostly in and near the fovea, which is dimpled, as you saw in that other figure. And the receptor fields tend to be relatively small. And they're quick, as the cones are quick. The rods are quite a bit smaller. So there's a real picture of the back of an eye, and there's this little dark area is dimpled. That's the fovea, and there's where your optic nerve leaves, which also creates a blind spot because if the axons of the eye have to leave, they leave down here. They have to pop through all these layers. And as they pop through all these layers, they push the receptor cells aside. So wherever, all day long, you have a blind spot in each eye and you don't see them because the nervous system spares you that fact unless you do these kinds of tests. And then you can see your blind spot, and it's weird. People who get migraines, by the way, sometimes get very big blind spots. I knew somebody who would look at me and say, "Oh, I'm getting a migraine because your head disappeared." She could see through my head. Of course, she wasn't through my head. Her brain was trying to, or her nervous system was trying to fill in for disruption of neural activity in cells relating to the fovea. And there's a discussion of different. The eye has evolved multiple times, and kind of funny that people say that a poorly formed eye is useless. It is not. I have poorly formed eyes. I need glasses. I'm still very happy to have my eyes. And even if you can just see dark and light, that's still useful. So there's how things are wired up. And as we said, and troubled medieval philosophers, things are reversed on each retina. And we have binocular vision where both eyes have overlapping views of reality. Our left eye doesn't just look to the left of us, which would be true if we were frogs, but it looks at the left world and the right world. But you can see that the left world and right world are parsed as they are sent to our thalamus. And by the time things get to our occipital cortex, our visual cortex, left is on the right side, right is on the left side. Cleverly done. Primary visual cortex, V1, good name for it. And what good are two eyes? You can do these little tricks to see why eyes help us see the world in three dimensions. When I was a child, we used to have Viewmasters. And I still remember just being blown away by this. So a Viewmaster, you have a portal for each eye, so each eye is looking at a different image, and you get these little photo gels. I remember Donald Duck and Mickey Mouse, and the perception of depth that you get out of these is amazing. If we were alive, I would ask you, have you ever used a Viewmaster? I recommend it. They're fun. And that's related to a lot of this discussion here, and binocular two eyes disparity, differences between the two eyes. And we're good at that. And there's convergence, where you have to torque your eyes in as things get closer. And you can try that one, and you can feel your eyes pulling in. And then there's different depth cues. There are monocular cues and binocular cues, and these are monocular. You only need one eye to infer that the triangle is farther away than the square, and the square is farther away than the circle. And here's another one. As you look at the real world, if we were in class, we would be looking at the carpet, and the texture of the carpet when it's near you is coarser and bigger. And of course, if I were looking out across the classroom, students sitting in the front would be bigger. And as you go further away, things get smaller, and things that are far away also seem to be higher. I don't know if you've ever noticed at sometimes a lake, but certainly the ocean, when you look out across the ocean, it looks higher, like it's at eye level, and that's relative height. And there's linear perspective, which we've seen a hint of from Aristotle again. This illusion is just blow you away. I mean, I teach this. I see what's in B every semester, and it's still hard to convince myself that the guy in the white shirt is the same size as the guy in the gray shirt. But it is. And motion parallax. If I were in the front of the classroom walking back and forth, people closer to me would be moving faster, and people farther away from me would be moving slower. And if you were driving cross country or sitting in a train, this would be very obvious. And there's a point off in the distance where the motion seems to stop. And beyond that point in the distance, objects start moving with you. Children report this accurately. They say the moon is following them, or the clouds are following them. They're accurately reporting what they're seeing. Hazing. I lived in LA. Oh my God, so much haze. And came back to Scranton, and it was very clear relative to Los Angeles. By the way, Scranton was one of the first places to have a serious climate issue with the smoke from all of our coal-fired heating units. And we're in a valley, and the wind didn't take the smoke away for a bit, and we had quite a bit of haze that day. And there's the motion parallax. And there's the pupil in the iris, shows up in that one. There was just a report that people with wider diameter irises are more intelligent. I'll have to look into that one. Nobody does real photography anymore. But how open your iris is affects depth of field. So with a pinpoint opening, you get a fairly deep field of focus. And here you would have a shallow field of focus. So if you're doing portrait photography, usually you want this. But if you're looking out across, a complex scene and you want everything in focus, you want this. Nobody knows this anymore because the camera, your cell phone does it for you. And we can see across very wide ranges of intensity. And of course, rods and cones are going to be important here too. And there's adaptation. Walking into a darkened theater. I walked into a darkened theater on a sunny Saturday afternoon, and I literally sat on someone. A little squeaking sound from behind me. I couldn't see them because I was still, light adapted. After I sat there for a while, I could see the person I had almost crushed. And then when you leave the theater, the light in the parking lot sometimes hurts your eyes because you're now dark adapted. By the way, how quickly we dark adapt is very nicely timed to how long it takes for darkness to fall on planet Earth, as if we evolved here. Rainbows, Isaac Newton, why things have different colors, what they absorb, what they reflect, photons of light. Now we're going to get into wavelengths and short. The rainbow, Roy Jabiv, red, orange, yellow, indigo, violet. Violet would be the higher frequencies. Red would be the low frequencies. The book focuses on the trichromatic theory of color vision. It's not the only thing that's going on with color vision, but you after images. Oh, there's a point process theory. Good, that's in there too. So you get lawful after images that are complementary. Yellow, blue, blue, yellow, red, green. And there's our photoreceptors. And I liked to sing the former Sesame Street song, one of these things is not like the other. And the thing that isn't like the other is the short cones, indigo, blue. Rods are sort of in the middle and they overlap with the other response functions. The medium cones overlap a lot. The red cones overlap a lot. Notice, by the way, that the rods do not go out as far as the long cones do, which is one of the reasons that red sports cars in dim illumination look black. By the way, the ancient Greeks, I think the Old Testament, they never mentioned blue. They never mentioned blue. Why is that? And there's colors, and right, to take shortcuts. These illusions are stunning. The brain is always, well, and by the way, the retina is an extended part of the brain. We're always working on sensory information, not just vision, to make sense of it. Shape constancy, size constancy, color constancies. We don't even think about them, but we rely on them every day. And there's the Ponzo illusion, and we saw that with the related guy lying on the tracks. And these color things are incredible. And to think that this is the same as that is amazing. And the same thing with this spot and that spot. Um, sensory systems uh, are dynamic. Gestalt, not Gestalt. Gestalt, Germans again. Rules of perception, there's a few of them there. Continuity, proximity, similarity, closure. Those Germans. And symmetry. And we flip back and forth between the two faces. And I think we're escaping vision and we're getting to the auditory system. And this is what I did my doctoral dissertation on. And vibrations in air, typically, water sometimes, but our auditory system clearly developed from things that lived in water like fish. And we do remarkably well listening to air with systems that evolved in the water. Pinna, I did my doctor's dissertation on the pinna reflex in part, and I can wiggle my ears. And when I wiggle my ears, I can also make a rumbling sound. And it's fun. We don't move our ears. Most people can't wiggle their ears, but and even those of us who can, it's subtle. But I did my research on rats, and I would go into the rat colony and go to them. And if you did it quietly, their little ears would wiggle. And if you made it too loud or they were afraid, they would startle. And I did my dissertation in part on that too. And tympanic membrane, the three smallest bones in your body, malleus, incus, and stapes, hammer, anvil, and stirrup, and then your inner ear with your vestibular system and cochlea. And there are two little muscles in there, which aren't named, but that rumbling sound I can make comes from one of those. And we have windows. We have an oval window, which the stapes sits on, and a round window that is membrane covered, but no bones are associated with it. So energy sent into the cochlea through the oval window can be pressure released through the round window typically without damaging hair cells. This is a little hard to get your head around. If you've ever eaten escargot or played much with snails, I used to do a lot of research with snails. They're coiled, and so is your cochlea, which is snail. And the high frequencies tend to affect the membrane that's depicted here, closer to the oval window and therefore stapes, and different frequencies moved down the cochlea, lower frequencies at the far end. And you can see some weird stuff here. The size of the chambers of the cochlea get bigger as you move away from the oval window, but that membrane, the green membrane, gets wider. Oh no, that's a bad picture. That's a bad picture. Actually, the chambers get smaller. It is true that the green membrane gets wider. The chamber gets smaller, the membrane gets wider. Ooh, Mark, you lied to me. So there's a discussion of the snail-shaped cochlea. Hair cells. Hair cells don't have axons. They synapse with sensory cells immediately without axons. And they do have little hairs, and vibrations in those hairs cause them to release or not release neurotransmitters. And don't want them destroyed. Largely, they don't come back. You go to recreational music and you come out of there with a hollow feeling in your head and you have a little trouble hearing, you probably have suffered some permanent hearing loss. Just a little bit, just a skosh. But if you're planning on living to be old, you should really try to minimize that. So localization of sound in space, usually done with binaural cues using, of course, both ears. And physics is important here. So there's a sound source, and you can think of the waves being like waves in the ocean, and there's this big island, which is your head. And when you are Captain Jack Sparrow and you want to anchor on that island, you want to anchor away from the wave patterns, where it's a lot calmer. And there's different theories, as there were with color vision, for how we do this. And the modern theory, the duplex theory, is we use both intensities and time of arrival. It depends. And boy, you don't see microseconds too often. Hair cells, binaural cues, sound shadowing, of course, is intensity. Time of arrival would be that the sound arrives at your proximal ear, in this case, the left ear first. By the way, if you've ever spent any time near the ocean and you've heard a foghorn, what frequency is it? Is it high frequency or low frequency? And the answer is it's low. Why? Because physical objects, low frequency sounds can usually just roll over them without casting much of a sound shadow, but you still have time of arrival. If foghorns were high frequency and a ship got between you and the foghorn, that would cast a very big sound shadow. And of course, how well you cast sound shadows is an interaction between how big you are, and we're pretty big, and the frequency of the sound. Mice and rats are quite a bit smaller. And taste and smell. We're good at taste and smell. And 5 taste receptors contribute to flavor. And flavor is the tastant. And boy, they hire psychologists in the food industry to make snacks. You can't help but keep eating. So there's, the taste buds and little receptors, and you can see that they don't have axons either. And they have a cruel life. They don't last long. They have to be replaced. And there's discussion of sweet, sour, salty, and bitter, and what the effective cues for those are down here, and why is bitter important to us. A lot of toxic substances are bitter. And of course, sugar is important to us because in our evolutionary history, we needed calories. We didn't have to worry about prom season or beach season and remaining slim. You had to pack in some calories, and sweet foods usually contribute a fair amount to that. And umami is interesting, savory. Uh, and there's no denying that we evolved eating the flesh of animals. So there's your tongue. And the book points out a myth that, the miracle fruit, yeah. There's a myth, I was down here. The myth is here somewhere. That different parts of the tongue are differentially sensitive uh, to different tastes. In fact, in our education department here, I saw that graph. It's not true. It's not true. Uh, you can taste virtually everything everywhere. It's a myth. Oh, this trust me. It's a myth. And there's our olfactory system. And compared to dogs, this is pretty puny. But notice what goes on here. You have receptor neurons that have to send their axons through little channels in your bones up to the olfactory bulb. And some people, when they get a serious whack to the head, the brain moves. And of course, this doesn't, and the axons are severed. And you can have hopefully short-term anosmia after that. Things often grow back, but they don't always grow back. One of my students was anosmic, and he went to work for a while at the world's most famous chemical sensory research facility in Philadelphia, and he became part of a study because they found out he couldn't smell and they wanted to know why. Um, Smell is, and here's COVID again. Smell and taste uh, can be severely messed up by COVID, and sometimes it takes a very long time for things to come back. And there's bionic eyes with the array of electrodes right on the retina. Yipes. Um, okay. Oh my God, it's over. So that was kind of fun. Let me think back to see if there's anything that I wanted to say that I didn't. I told you about the myth. It's in the book. I just forget exactly where it was. And in terms of aging, I mentioned in the last chapter, these neurons have to be replaced, neurogenesis. And if you're my age and you start not being able to smell things very well, that may not be a good sign for your longevity, independent of COVID. So that whole figure is contradicting the popular myth, every part of your tongue detects all five tastes. Yes. So the myth must have been on the other page. Not important where it is in the text. It is in the text. Let me just flip back a little bit. I know it went quickly because there was a ton of stuff in here. I love sensational reception. If I'm ever back in the classroom, I recommend it. I have good auditory. stories. So when you unroll a snail, the fat part of the snail would be here, and the thin part of the snail would be there. But that green membrane does get wider as you go through the cochlea. Beautiful pictures of the middle and inner ears and sound. Okay, I'm happy. So thanks for stopping by. We're almost through week 2.